Hi, my name is Susan Cohen, and today we are here in the Golden Lab at UC San Diego. This video is going to introduce you to the use of luciferase reporters in cyanobacteria in order to monitor their circadian rhythms. Cyanobacteria are a phylum of bacteria that obtain their energy via photosynthesis. Cyanobacteria are also special in that they display circadian rhythms of gene expression, with rhythmic expression of up to 80% of their genomes is observed. A better understanding of the circadian mechanisms of these simple bacteria could lend great knowledge and insight into the evolutionary basis of circadian rhythms and the means by which cells can tell time. In order to visualize the cycling of rhythmically expressed genes, we will first insert a reporter into the genome. In this case, we are going to use luciferase. The species of cyanobacteria we are using today is Synechococcus elongatus PCC7942, which can be easily transformed, meaning that the cells readily take up exogenous DNA. So first, we have to obtain the genes that code for the luciferase enzyme, as well as its substrate, luciferin, from another bacterial species. These genes are cloned into a plasmid that will allow them to be expressed in S. elongatus, and driven by a promoter sequence of interest. The promoter sequence chosen will dictate when the genes are expressed. This will lead to the production of the luciferase enzyme that will react with the luciferin substrate to produce light, which we can then measure. The plasmid is placed in the liquid media with the cyanobacteria, which can take up the DNA and integrate it into their genome. Along with the luciferase and luciferin genes, we also include a gene that creates resistance to a specific antibiotic. This allows us to culture the bacteria on media treated with the antibiotic, and then only cells that successfully integrate the DNA we have given them will survive, because the antibiotic will kill any cells that did not get the antibiotic resistance cassette. Once we have the cells that contain the antibiotic resistance gene, and therefore the luciferase-luciferin construct, we will move them from a plate into our experimental setup. At this point, we may make other genetic modifications, such as decreasing or increasing expression of particular genes to observe how these alterations affect circadian expression of the luciferase reporter. Today, I will show you 4 different samples. First, an unaltered or wild-type sample with its clock genes intact. Second, an arrhythmic mutant. Third, a long-period mutant. And fourth, a short-period mutant. Note that all of these samples came from the same S. elongatus species that had the luciferase-luciferin genes transformed. From this strain, other modifications to the clock genes were introduced, giving us the different mutants that we will observe today. When the reporter gene is expressed, we will be able to visualize expression by observing the amount of light produced by the cells. But first, we need to entrain the bacteria to a light-dark cycle so that all of the bacteria have their clocks set to the same time. In their natural habitat, the bacteria would entrain to the environmental cycle driven by the rising and setting of the sun. In the lab, we use incubators with time lights to simulate the environment. This also gives us the flexibility to plan our experiments without having to come in at dawn. Once the bacteria of interest are inoculated onto a plate where they will grow and be measured for the duration of the experiment, they are placed in the incubator and left to entrain to a light-dark cycle with lights on for 12 hours and lights off for 12 hours, otherwise known as LD1212.
Hi, I'm Ryan Schulteberger, a postdoc at UC San Diego, and I'm going to walk you through how to gather the data from the cyanobacterial samples that Susan prepared. The first thing we need to do is take our entrained bacteria to an imaging station where we can visualize the luciferase. Notice that our camera setup is in a 30 degrees Celsius warm room. This is to ensure that the temperature is constant throughout our experiment and that the growth of the bacteria will not be hindered. Agar plates containing our samples will be placed on the turntable, where cutouts have been made to specifically hold the plates. 9 plates can be accommodated during each experiment. Overhead, you will notice a light source that will keep our samples in constant lighted conditions for the duration of the experiment. This is important because cyanobacteria require light to continue growing. Once every 2 hours, the table will rotate each plate under a CCD camera to be imaged. You can see that the plate that is being imaged by the camera is covered and is kept in the dark so the weak amount of light produced by the bacteria can be detected by the camera. This is important because otherwise, the camera will measure light from the external light source, which we have placed over the bacteria, and the bioluminescent signal will be lost to ambient light noise. The CCD camera will detect the amount of photons, light, produced over a 3-minute period of time. After imaging, the turntable will rotate to release the plate back into the light. Note that we determined experimentally that these short periods of darkness every 2 hours do not affect the clock. The cells can reset their clocks if presented with hours of darkness, and the rhythms can get messy if we give a short dark pulse too frequently. Both the data and the video are collected by the computer. We can then use the data we have acquired to visualize and analyze the activity of the luciferase in the bacteria using specialized software.
Now you will learn how to take raw data from an experiment and turn it into a graph that displays the luciferase activity from the cells over multiple days. Let's take a look at the video we collected from 7 days worth of data, taking note that the video has been sped up for convenient viewing. We can see that our wild-type cells, highlighted in blue, have an approximately 24-hour rhythm of gene expression that we are monitoring by measuring bioluminescence. Highlighted in red, we can see a strain lacking the core oscillator that has no rhythm, or is arrhythmic. In orange, you can see a long-period mutant, or a strain with a genetic alteration that causes the clock to run longer than 24 hours. In green, we can see a short-period mutant, or a strain that harbors a mutation that causes the clock to run faster than 24 hours. Now that we have acquired images of our cyanobacteria, we can obtain quantitative data that measure the rhythmicity of expression of the reporter gene. The CCD camera we are using is able to detect the light emission by measuring the average pixel intensity of each strain over time. This allows the program to clearly show expression of genes over a selected time period, as you can see on this graph. As the colony gets brighter, the amount of light detected increases. Then as the colony becomes dim, the amount of light detected decreases. This pattern repeats each day as the bacteria follow their cycle. Let's take a look at the graph in more detail. Starting with the wild-type data, on the y-axis we have the amount of bioluminescence being produced by the bacteria measured as pixel intensity. On the x-axis we have time in constant light, also known as LL. After the bacteria have been entrained to 2 cycles of light-dark, or LD 12-12, 12 hours in the light and 12 hours in the dark. Here we can see the blue line corresponds to the wild-type data, red to the arrhythmic mutant, green to the short-period mutant, and orange to the long-period mutant. We can see deviations in rhythmicity if we observe the peaks in the wild-type strain and compare them to that of the mutant. The short-period mutant, which runs slightly faster, peaks earlier or to the left of the wild-type. Conversely, if we look at the long-period mutant, we can see that it runs slightly longer and peaks to the right of the wild-type. We hope that this laboratory demonstration was useful to you and that you now have a better idea of how circadian biologists go about obtaining and interpreting data of circadian rhythms in cyanobacteria.